Okay, so last time I left off talking about ELISAs, the EIA enzyme immunoassay, and radioimmunoassays, which are all sort of similar to each other, just different variants on the same theme. Um, I'll pick up today with Western blots. And a Western blot is basically a test for antibodies or antigens. Usually you're looking for antibodies. And you can look for multiple different antibodies or antigens at the same time. There's almost no limit to how many you can find. And there's three major steps to a Western blot. First, you run an SDS page on your antigen. Typically, you have a known antigen that you add to the SDS page analysis. So the SDS page gel looks like this. It's actually vertical, and it runs down toward the positive end after you treat it with beta-mercaptoethanol and SDS, right? Giving it a negative charge, it moves toward the positive end. Then you transfer your antigen proteins onto a paper-like substance, either PVDF or nitrocellulose. And then last, you add the antibody, and then you analyze whether the antibody is present. And the results look something like this, which you can see here. So this is a really nice depiction of how this test works. So step one is the SDS page in the Western blot, and you're doing the SDS page on an antigen. Remember, SDS page gels are used for proteins. Agarose gels are used usually for amino acid, or excuse me, agarose gels are usually used for DNA, although they can technically also be used for proteins. But so first you're going to combine your antigen with a loading dye. And the loading dye contains a substance called beta-mercaptoethanol. And this is an agent that reduces disulfide bonds back to sulfhydryl groups. And that denatures the proteins and makes it all kind of fair. So it turns your protein into a linear string of amino acids, basically, that is not folded whatsoever. So it reduces it to primary structure. Then you boil your antigen for 5 minutes. And that is enough to basically disrupt all intramolecular forces and force the protein to be linear. And also you're going to separate any subunits of your protein into multiple different bands in the Western. So any multi-subunit proteins will be separated into multiple bands of different sizes. So only linear epitopes will be recognized and assessed in a Western blot. That's usually not an issue because antibodies are going to be made to pretty much any epitope or derivation thereof of any antigen. Okay, so if you were to immunize a mouse with an intact protein that was multi-subunit, it wouldn't only make antibodies against the 3D epitopes, it would also make them against the linear epitopes. So you will have antibody if you've been exposed to this antigen before, or if your serum individual has been exposed to this antigen before. Then you're going to run the SDS page once you have denatured the antigen and reduced the disulfide bonds. And this stands for sodium dodecyl sulfate. That is the substance that you treat the proteins with, and it's it confers a negative charge to them, and it also suspends them in like a soapy sort of solution. And then the page stands for polyacrylamide gel electrophoresis, and that is just the gel technique. It's made of polyacrylamide. The SDS page separates antigen proteins based entirely on molecular mass. They contain a uniform negative charge when you add the SDS, and then they run toward the positive end of the field, which is on the far side. Larger proteins stay toward the top of the gel, and smaller proteins move more quickly. They move toward the bottom of the gel. And then you also add a ladder to serve as a standard so that you can judge the mass and the amount of proteins that you have in the sample. This actually generates like a lot of heat, so you usually do a Western blot inside a refrigerator, weirdly enough. So you basically load your samples into all the different wells, and you also have them combined with the loading dye that contains the SDS, or excuse me, that contains the beta-mercaptoethanol. And the loading dye also helps them to sink a little bit and to stay in the well. Then you turn on the electricity, they run through the gel. Your ladder is like a comparison reference point. And then usually each well might contain like a serial dilution, or it might contain serum from different individuals, um, or it might just be like you're running the same thing multiple times and you want to make sure that you capture your results consistently. Okay, so you've run them through. The only issue with doing the Western blot, with doing the SDS phage, is you can't actually see the bands, right? You can't actually see them. They are invisible. Now, normally, if you were to only do an SDS phage, where you're separating proteins based on molecular mass, what you would normally do in a normal SDS phage is you would add this dye called Kamasi blue, and that would stain your proteins blue, right? But if you do this during a Western, this changes the protein so that the antibody won't be able to bind. So you don't actually add the Kamasi blue during a Western blot. So you actually stop at the point of where they're invisible, but they've run through the gel. Okay, so you do not add your Kamasi blue dye during a Western blot. Okay, what you instead do after your SDS phage, and this is just showing you another view of an SDS phage. What you do after you've run the proteins through the SDS phage is you transfer the proteins onto nitrocellulose or PVDF. And the reason you're going to do this is because when they're in the polyacrylamide gel, the antibodies can't get into that. They can't get into the gel. So the proteins are inaccessible in the polyacrylamide. So you have to transfer it onto this sort of flat paper membrane, and you do that in a transfer buffer. You create this hamburger patty, basically. You have a support grid, then a pad, then the filter paper, and then you add your PVDF or your nitrocellulose, your transfer membrane, and then you put your gel beneath that, and then you put another filter paper, another pad, and then another support grid. So you make up this big hamburger patty. You assemble the gel and membrane sandwich, and then you basically press them together, and then you start the transfer. The transfer is again vertical, but in this case, the negative charge side is on the side of the gel, and then the positive side is on the side of the transfer membrane, the PVDF or the nitrocellulose. So they actually migrate onto the paper using an electric field. But instead of migrating down, they're now migrating to the right here, relative to this image. Okay. So you run the separated protein mixture on another electric field. This time the positive end is behind the paper, the PVDF or nitrocellulose transfer membrane. Antibody cannot bind to proteins inside of an SDS phage gel, so you need to transfer them onto basically paper, I guess expensive paper, where antibodies can bind to. Polyvinylidene fluoride is PVDF, and then nitrocellulose, which is sometimes abbreviated NC. They are just inert polymers that antibodies can get to the antigen on. So you're trying to get your antigen available for antibody binding. Okay. The only thing is these are actually all very toxic. The polyamide is toxic, the nitrocellulose is toxic, the PVDF is somewhat toxic, so you want to wear gloves when you're doing this. But at the end of the day, when you do this, you've now migrated your antigen proteins onto PVDF or nitrocellulose. So you have them on a film where they're exposed and available for antibody binding. However, you need to block the antigen after this, okay? So once you've, ran your hamburger and you've let it incubate for an hour or so, you block your antigen and then you add your antibody. Now, what you need to do, the PVDF and the nitrocellulose, and probably also the antigen, all of them have somewhat of an ability to bind to antibody, right? So what you want to do is make it harder for antibodies to bind, and that is called blocking. So you want to block non-specific interactions between the antibody and the antigen. The way you do that is you use TBS tween gelatin, or you can also use TBS tween with powdered milk, which is like another inert substance. that will block antibody binding. So TBS tween gelatin, what is it? It's tris buffered saline and polysorbate 20, tween 20, with gelatin, just like you make Jello out of. The TBS tween is just an inert solution where proteins are happy and they're in their native conformation. But the gelatin is the blocking agent, okay? So the blocking agent is going to alter, not alert, but alter the delta G that is necessary for binding to occur. So basically, the binding has to be very specific for it to occur in the presence of gelatin. Okay, so the antibody has to be very specific for the antigen in order to bind across this gelatin blocking agent. Gelatin and powdered milk both block ionic interactions specifically, and that just makes it so that you need a more negative delta G in order for the binding to occur, essentially. So specific antigen-antibody interactions are not really phased because remember those have some of the tightest binding affinities of like any protein that we know of. They're very specific, very good at binding to each other. But the non-specific binding that you'll see is blocked and it's inhibited almost entirely. So this just reduces the background signal when you use the gelatin. Okay. So the whole point of adding the antibody, or what you're doing in step 3, is adding the antibody. So you're adding it to your PVDF or your nitrocellulose. So if it can, the antibody will bind to its target antigen on the paper, and you just incubate the antibody or the serum sample by you incubate it with the PVDF or nitrocellulose in a tube containing TBS tween gelatin, and then you just shake it on like a lightweight shaker with like, I don't know, an RPM for every like 5 seconds. So it's like a slowly shaking shaker. And they just incubate there for like an hour or two. It takes a good while, so more like 2 hours. And the antibody will eventually bind to the antigen, and the non-specific binding will not occur because of the gelatin or the powdered milk that you're using. Next, you're going to add tris PBS buffer to remove all non-specific antibodies from the serum mixture or from whatever you're adding, usually serum, and you're looking for an antibody. So now you have immune complexes present on the PVDF because it's like an antigen bound to the PVDF, and the antigen is also bound to an antibody. Next, you add a secondary antibody, and the secondary is linked to alkaline phosphatase or horseradish peroxidase, some kind of enzyme that will take a substrate and change it into a colored chromogen. And you're going to look for a color change, and anywhere there's a color change, there was an antigen that had specific antibody for it in the serum sample. So the mixture was positive for your antibody. You're usually looking for antibody in a Western, but you could look for antigen potentially using, like a known mixture of antibodies. Alternatively, you might use a radioactively labeled secondary antibody. No one really does that because enzymes are way more safe and they're also cheaper, but they're less sensitive is the only thing. So radioactivity is more sensitive, it's just more dangerous and more expensive. So this is what your results look like. Here we have a control strip where no antigen was found, and that was treated with the serum sample from mice. Here, or I guess I'll actually start over here. Here we have like pretty concentrated serum sample that was added to the antigen containing paper, and these are all PVDF strips. So this is like a 1 to 30 dilution of serum to TBS tween gelatin, and you can see anywhere there's dark bands, that is a positive antibody antigen interaction. Okay, so this is like what the Western looks like when you're done with it. You usually cut it into strips so that you can, I don't know, use your reagents more efficiently, because PVDF comes in these like big sheets of paper. You cut those into strips for this purpose. Okay. So yeah, this is basically what the Western looks like at the end of the day. Anywhere you see dark spots is is a positive reaction for that antigen-antibody interaction. Okay. And you can see that you can assess for multiple antigens here and multiple antibodies here. So they actually do this for the diagnosis of HIV, or at least they used to. I think they just do an ELISA now. They do this for the diagnosis of certain diseases that have a variable antibody response, because this allows you to assess a lot of different antigen-antibody interactions. So it can be really useful for that purpose. So any questions on the Western? Going once, going twice. Okay. In some cases, you want to get your antigen or antibody back. And when you want to do that, you perform this thing called stripping your Western blot. Okay. For example, you might not have very much antigen or very much antibody, so you might want to get it back. So what you're going to do is you heat the PVDF or nitrocellulose, you add beta-mercaptoethanol, and you incubate in a stripping buffer that contains TBS and SDS, and then you rinse with water and rinse with TBST. Then you reblock, and your antigen and antibodies are resuspended in solution. Good luck separating them, but you can actually strip the blot, so that can be useful. I'm actually not 100% sure how you would separate the two, because I don't know, you kind of just have everything all floating around in solution at that point, and it's going to be hard to isolate. Okay, and then there's monoclonal antibodies, which are kind of confusing because they're very involved to produce. But these are actually used for medications. There's like hundreds of these on the market. And so they are very important immunological developments, and this is like the way of the future. These were invented in the 80s, mid 80s to early 90s. So they're not exactly new, but these are being constantly developed. There was just a new Alzheimer's treatment that was a monoclonal antibody produced within the last year, and that just got FDA approval. So these are still being developed, to a very great extent. So monoclonal antibodies are basically antibodies of a single specificity and a single class made in the lab by a pure cell line, a genetically pure cell line of hybridoma plasma cells. So hybridoma, basically you take a myeloma cell and you fuse it physically with a plasma cell that is making antibody that you are interested in making a bunch of. Okay, so a myeloma cell is a cancer of plasma cell origin. So here's how it sort of works. You immunize typically a mouse with an antigen that you want to make antibody against. You want to make a monoclonal antibody against this. For example, they've made COVID treatments of monoclonal antibodies, and COVID is caused by the SARS-CoV-2 virus. So they take SARS-CoV-2 virus and they inject a mouse with it, probably a killed strain of the COVID virus. And then the mouse makes plasma cells that are making specific antibodies against the COVID-19 virus, or SARS-CoV-2. Those plasma cells have some issues. Plasma cells are short-lived. Okay? They live only for like 3 to 5 days, and then they die. But they also have some things that we are very interested in. We're very interested in making antibodies, right? And plasma cells make tons and tons of antibodies. So what we can do is fuse the plasma cells with a tumor cell, in this case myeloma cells, and we do that in PEG. We fuse their membranes physically together to create a hybridoma strain. Why do we use a tumor cell? Because a tumor cell is immortal. It never dies. So when you make a hybridoma, some of them, at least some of them, you only need really one cell to successfully come out of this process. It's not very efficient, but as long as you get one cell out of this, one successful hybridoma cell, you are good forever, because hybridoma cells are immortal. Now you have a cell that never dies. It replicates efficiently. It replicates eternally. All you have to do is change the medium and they won't die. Basically, you just give them new nutrients every 3 days or so, and they make tons of antibody for you. And you just grow these cells for as long as you want. And every few days you harvest the supernatant that contains the antibody that you want, and then you replace the supernatant with fresh nutrients, and you keep those hybridoma cells alive forever. This is actually done in these giant bioreactors that are like 1,000 gallons large. So I don't know if you ever played a video game and you saw those giant tanks. It looks something like that, but it's just literally like billions of hybridoma cells inside this huge vat. And they just change the media every 3 days to every 7 days, and those cells are pretty good. These antibodies are going to have the same affinity as the plasma cells that you start with, and generally they're going to be of the IgG class, and you're going to make sure that they're IgG before you choose your hybridoma cells that you want to keep and which ones you want to deposit into the waste. So the reason that you use myeloma cells is because they are from plasma cell origin, so they are very structurally similar to plasma cells. So the fusion is most likely to work if you do a myeloma cell line. Just because you're fusing membranes, and that's not a very efficient thing to do. Most of them are going to die when you do that. But if you use myeloma cells, you get the most efficient transformation, the most efficient fusion into a hybridoma cell. So here is a rundown of it. B cells die in 5 days in tissue culture, at the longest, right? So you can't keep a plasma cell alive for very long. This should really say plasma cells. But you really want that plasma cell to stay alive. That's your goal. You really want that plasma cell because it's making your monoclonal antibody. However, you know that myeloma cells never die in cell culture. They never die in cell culture, but if you place them into HAT media, which is hypoxanthine aminoterrin thymidine media, they do die. The thing is, B cells are resistant to the HAT media because they can do this really cool salvage pathway where they take the hypoxanthine and they convert it into the precursors for the purine synthesis. Okay, so the HAT media is a weird thing, but this is what you use to select between hybridoma cells and myeloma cells. So you just add all of your potential fusion results into HAT media, and then you wait a week. Okay. Here's what happens while you're waiting that week for them to grow up in the HAT media. Aminoterrin within the HAT media is an inhibitor, a very strong inhibitor of the enzyme dihydrofolate reductase. Dihydrofolate reductase normally takes a folic acid precursor compound called DHF, and it converts it into THF, which is the active version of folic acid. And the cell uses folic acid to turn certain substrates into purines for DNA and RNA synthesis. Okay, so THF is required in the normal pathway for purine synthesis of DNA and RNA. Okay, with aminoterrin present, this is not available. THF is no longer available because dihydrofolate reductase is dead. It's not working because it's being inhibited by aminoterrin. So you don't get the normal pathway of synthesizing DNA or RNA. Okay, so you can't do the normal way. So dihydrofolate cannot make THF for purine bases for RNA and DNA synthesis. However, this only kills myeloma cells. It does not kill B cells. Why does it not kill B cells? B cells can make use of hypoxanthine within the HAT medium to synthesize their purine bases. They have an enzyme called hypoxanthine guanine phosphoribosyltransferase, HGPRT or HPRT. Okay, so they have this weird enzyme that can take hypoxanthine and convert it into inositol monophosphate, which is the precursor for purine synthesis. Normally, that IMP, inositol monophosphate, can only be made in this normal pathway. But B cells and plasma cells have this really fancy pathway they can do where they take hypoxanthine and make it into the precursor for purine synthesis. This is called the salvage pathway or the scavenger pathway of purine synthesis. Myeloma cells do not have this enzyme. That is just a fact of nature. Myeloma cells don't have HGPRT, so they can't do salvage pathway synthesis. So while you're waiting those 7 days, um, these myeloma cells are going to die because they can't make DNA or RNA. So they die within like a day or two, because you need RNA for the cell to survive, right? You have to constantly be turning over your proteins and such. So they're going to die in a few days. But plasma cells, remember, they die after 3 to 5 days. So they're not going to live either if you wait a week. However, hybridoma cells, your hybrids of the myeloma and the plasma cells, the hybridoma cells, they are immortal, so they last forever, and they don't die in the HAT media because they have the HGPRT enzyme from their plasma cell fusion cell. They also make the monoclonal antibody that you're interested in. So all you have to do is immunize a mouse, harvest the plasma cells, try to fuse the plasma cells with myeloma cells, and then throw those fusion cell products into HAT media for 7 days, and then you come back in 7 days and you look for a cell under the microscope. Sometimes you might have a couple of cells in a well, sometimes you might have one. If there's a cell still alive in your well, then you have made a hybridoma cell that is immortal and hopefully makes your antibody. So once you do that, you're going to have to make sure that the antibody is directed against your antigen of interest, right? So here's how the fusion works. You use polyethylene glycol, PEG, to merge the membranes of your plasma cells and myeloma cells. Okay, and you're using myeloma cells because they're structurally similar to plasma cells. They have very similar organelle structures, so the membranes are just more compatible. The cell is less likely to die if you use myeloma cells. PEG is used to merge the membrane of the two cells. Relatively non-toxic, except in high levels, but you're going to use pretty high levels in this case. So this process is pretty messy, right? It's going to kill a lot of cells. Most of them are not going to succeed, but you just need at least one successful cell that is making antibody against your target, and then you are golden. You can make millions of dollars off of these treatments. So 2 figures for your enjoyment. This is like an old figure that I actually really like. You take the antigen, you immunize the mouse with it, you harvest the spleen and usually the bone marrow. And those spleen cells are HAT resistant because they're plasma cells. They have the HGPRT enzyme. And then you also take non-secreting myeloma cells that don't make antibody, and they are sensitive to HAT. And you fuse them in PEG. Then you culture them in HAT for 7 days. Okay, in these big, you're going to make like probably 100 of these plates that have 96 wells each. So this takes like tons and tons of pipetting, but you can make millions of dollars if you do this correctly. So the myeloma cells, during the 7 days of incubation in these plates, they are killed by the HAT media. The spleen cells die off after 3 to 5 days because plasma cells are not immortal, they die in culture. Only the fused cells survive. They survive both the HAT medium because they have partial plasma cell characteristic, and they're immortal because they have partial myeloma cell characteristic. Next, you're going to test for the antibodies in the supernatants of each of these wells. Okay, and if your antibody is being made in that well, guess what? You're going to clone the cell in that well, and you're going to make as many of that cell as you possibly can so that you can make a bunch of antibody, a bunch of monoclonal antibody. So you're going to have a bunch of clones of this hybridoma cell secreting an antibody of a single specificity and a single isotype, and then you're going to use that antibody that you get from the supernatant as a treatment for some disease or as a diagnostic tool, like for use in ELISA or an EIA. This is literally what they do to make these things. They literally make these myeloma cells, these hybridomas. And then there's the Janeway figure here as well. So the spleen cells are producing antibody from the mouse immunized with antigen A. But spleen cells are, they're polyclonal. Each cell makes a different antibody. You're only interested in this yellow one because this yellow one makes antibody for antigen. But you don't know which of them are making your antibody yet. And you fuse the cells with myeloma cells in PEG. You transfer to HAT medium. You wait 7 days. And then you you test each well. After 7 days, you test each well for 1 live cells. That tells you that you have hybridoma cell that successfully fused, right? And didn't die like all these unfortunate casualties. So you have a hybridoma cell making antibody, but that antibody has to be specific for the antigen that you want it to make antibody for. So you test it for reactivity with antigen A, and you can do that using an ELISA. You can do that using a precipitin assay. You can do that with a Western blot. You can do however you want to test for the antibody. You got to test for it. Okay. And then once you figure out which cells are making the antibody that you want them to make, you clone them and make millions and millions or billions of them in giant bioreactors. Okay, and they are immortal, and they also make the antibody of interest. And they make a single isotype and a single specificity of that antibody, right? So now you have a monoclonal antibody that you can harvest in very high levels. You can concentrate it, you can affinity purify it, and then turn it into an injectable medication. Or you can turn it into a diagnostic agent. Okay, so these are how like ELISA, like for example, in the quantiferon gold tuberculosis test, this is how your interferon gamma antibodies are made. They're made in a mouse organism, and then they have myeloma cells for these, or hybridoma cells for these. Okay. So any antibody you can buy on the market is made by a hybridoma cell, and this whole procedure has been gone through. Now this is very time consuming and it's pretty expensive, but if you manage to make something that actually works for its intended purpose, you can charge a lot of money for it, and believe me, they charge a lot of money for it. We're talking multiple $1,000s per injection, which is pretty messed up. But people get really rich on this. You probably shouldn't charge $1,000 for a medication that saves lives, in my opinion. But you know, people do it, and they make lots and lots of money. They're very specialized, advanced medicines, and this is certainly the way of the future. So this is a summary table of the positives of each of the cell types. Plasma cells, they only live 3 to 5 days, but myeloma cells are immortal. Plasma cells have the HPRT enzyme, and so they can make purines. Myeloma cells do not, they die. Plasma cells don't succumb to the HAP medium, and they do make the monoclonal antibody for you, whereas myeloma cells die in the HAP medium and they don't make antibody. Hybridoma cells have the positives of both of those cell lines. So they live forever, they don't die in the HAP medium, and they also make antibody against your antigen of interest. However, you have to make sure that their antibody that they're making is actually specific for your antigen, okay? People do this like on mass scales. They take lots of mice and they make lots of, you know, hybridoma cells, and they can make an antibody that is directed against a very specific epitope that they want to target. This is why there are antibodies directed against the receptor binding domain of the spike protein of the COVID-19 virus, because they have made tons and tons of hybridoma cells and only cloned the ones that are making that antibody. Okay. So remember, you have to select for the antibody with the desired specificity, and you're only going to clone the cells that are making that antibody. Okay. So that is monoclonal antibodies, and I still don't hear any questions. So feel free to interrupt me at whatever point. You can raise your hand, I'll hear it, or you can just unmute yourself and I'll hear it. And then we have to talk about phage display libraries, which are equally complicated but a little more molecular than the monoclonal antibodies. So this is how it works. This is in word form, at least. First, you immunize mice with an antigen that you want to make an antibody against. And you're really not making an antibody, you're making a tiny little piece of an antibody. That is your goal in this process. Okay, so you're going to immunize mice with an antigen of interest. B cells are going to be formed in that mouse that make antibody specific for your antigen. Next, you're going to harvest the bone marrow and spleen because that's where all the B cells are. That's where all the plasma cells are. You're going to take the cells from the bone marrow and the spleen and you're going to lyse them with a lysing buffer. Then you will use reverse transcriptase to turn the cell's mRNA into cDNA, complementary DNA. So you have a bunch of DNA molecules that came from mRNA molecules in the cell. Now, some of those mRNA molecules were going to encode an antibody. Some of them were going to encode cell survival proteins. Some of them were going to encode Krebs cycle proteins. You don't know what all of your mRNA molecules were going to encode, right? So you really have like a bunch of cDNAs. You're only interested in some of them, though. Interested in the ones that encode an antibody fragment. Okay? Remember, the heavy chain and light chain are made as separate mRNAs. So you're going to need a heavy and a light chain. So what you do is you run a PCR on your cDNA library that contains a bunch of different cDNA molecules from the mRNA of the cells. Your primers in the PCR are only going to replicate the FAB regions of the antibody. The primers also contain restriction enzyme cutting sites that can allow you to turn the DNA into a circular plasmid-like molecule. Okay? So you incorporate your DNA into a T4 or an M13 bacteriophage using restriction enzymes that were encoded for by your primer. Okay. You can use really any bacteriophage for this. Use any of the T's or M's. Then you're going to infect E. coli cells with the bacteriophages to allow them to replicate. Now you have a bunch of T4 phages. Now, because those T4 phages contained the pieces of DNA that encoded an antibody, those T4 phages have the FAB region of the antibody on the external surface of their protein coat. Okay? So instead of having like a cell surface binding protein on their coat, they now have an antibody on their coat. They have a little piece of an antibody called a single chain fragment variable piece. Okay? Now, like I said, some of those DNA pieces were not encoding an antibody, right? So your PCR is only going to amplify the pieces of DNA that encoded an antibody. So you know that all of the genes you're working with at least encode an antibody of some kind. However, in the spleen, you have tons of B cells, you have tons of plasma cells. And all of those B cells and plasma cells, they might be making antibody against your antigen of interest, but they also might be making antibody against anything else. They might be making antibody against literally anything. So you need to make sure that the antibodies on the phage coat are actually specific for your antigen. So you do this process called panning. This is where you basically do sort of like an ELISA, where you look for the correct specificity using antigen bound to a plate. Okay? And as long as your phages are bound to your antigen on your plate, you know that they are making antibodies specific for, or that they have a piece of an antibody that is specific for your antigen. So now you have all these phages that contain little pieces of an antibody that binds to an antigen. Why does that matter? Well, you can actually cut off that little antibody piece using a protease called papain. And so you can chop off your SCFV piece from the phages. And that gives you a little tiny piece of an antibody that is so small and tiny that it can get into like anywhere in the human body. And it can bind to very small targets and do things that antibodies cannot do. And that's really the utility of this this whole thing, is you have a tiny variant of an antibody that can bind to stuff that normal antibodies cannot bind to. And you can also make them in mass, kind of like a monoclonal antibody. So here's what it looks like. You basically are interested in an antibody protein, but you only want the variable region, right? You only want the antibody binding domain. So what you're going to do is you're going to take the gene for the variable region of the heavy chain and the gene for the variable region of the light chain, and you're going to amplify those in a PCR after you lyse the cells. Then you're going to construct a fusion protein of the V region of both the heavy and light chain, and you're going to link the heavy chain and the light chain together with a little bit of linker DNA in between, as well as a promoter region. This isn't the promoter region. You're going to place it on the phage genome right next to basically the top of the phage. Okay, so now the top of the bacteriophage contains little antibody pieces linked to what would normally be found there. You're only interested in the antibody that binds to the red antigen, though, so you're only going to want to keep the phages that have the red antibody pieces, right? You want to get rid of everything else because those are antibody fragments that don't bind to your antigen of interest. So you're going to have to pan or select for phages with the desired V regions by specifically binding them to an antigen on a plate. Okay, so you coat a plate with antigen, and then you add all your different phages, and the ones that remain bound after the wash step are the ones that you're interested in. You're going to elute those, and then you can grow them up inside more E. coli cells, or you can off from the phage and then use them to make drugs or to make diagnostics. Usually you're interested in making drugs that can target very tiny antigens, and you can also use this for like antibody delivered drug therapy. For example, you might be interested in making phages against cancer, a certain type of cancer, right? And you know that this cancer type expresses a weird protein on its surface that isn't found on other cells. So what you're going to do, or maybe it expresses like a really high amount of that protein on its surface, whereas other cells don't express nearly as much of that protein. So you're going to immunize your mice with that protein, that tumor antigen, that cancer antigen, and then you're going to do this whole process, right? And they're going to make antibodies against a variety of things, but you're only interested in antibodies that bind to the cancer antigen. So you're going to pan them using the cancer antigen bound to the plate. Now you have a little tiny antibody fragment that binds to basically a tumor antigen, a tumor-associated antigen, and you can actually bind those little pieces of an antibody to like a cancer drug, like etoposide or whatever cancer drug you think can kill the cancer cells in question. So this can be used for immunotherapy of cancer. It can be used for a number of different things. Probably the most common use is in killing cancer cells. And it's going to, when you inject it into a patient, it's going to bind to the cancer cells, and you can conjugate a cancer drug to the scFv fragment that you've cut off of all those phages, and that antibody will come in, it will bind to the tumor antigen on the surface of the cancer cells, and then the drug will dissociate and kill the cell. So this is like antibody-targeted drug delivery. There's other techniques too. You can conjugate pretty much anything you want to these, as long as you think it might be toxic to the drug or to the cell that you're targeting it to. Okay, so there's a number of different uses for this. So how on earth are you going to do this PCR? There are areas of the V region and the light chain, well, the V region of the heavy and light chain, that are only a little bit variable, and then there's regions that are extremely variable from antibody to antibody. You want to amplify the V region of the heavy chain and the light chain, right? You want to amplify the V region of the heavy chain and the light chain. So what you're going to do is you're going to take a bunch of primers that bind down here and are directed this way, and then you're going to take primers that bind down here and are directed this way, and you're going to throw those into the PCR reactor. That way you can amplify basically any antibody that there might be present. You're going to do the same thing for the light chain. You're going to take an antibody that binds, or excuse me, you're going to take a primer that binds down here and is going this way, and you're going to take a primer that binds down here and is going this way. So you're going to flank the V region with primers, and you're going to probably use a lot of primers because these are still pretty variable. You're going to use a bunch of different primers that are known to bind down here and throw them into the PCR reactor so that you can amplify a bunch of V regions, okay? So these are called the framework regions of the antibody. These are less variable than the complementarity determining regions, the CDRs. Those are the ones that encode the actual paratope that binds to the epitope, okay? So these areas in red here are the hypervariable regions, also called the complementarity determining regions, or CDRs. Okay, so same thing. And you're going to try and flank those by using primers that bind to either end. That way you can amplify the V region. Okay, so this is what an antibody really looks like in the DNA that encodes the antibody. The antibody is basically nine beta sheets like this, arranged like this in a big polypeptide chain. N terminus here, C terminus here, a disulfide bond between this loop and this loop. You're going to want to amplify basically this whole protein fragment, but the DNA encoding that protein fragment. Okay, so you use primer that binds here, primer that binds here. You're going to amplify this sequence, all of these sequences. Okay, and that gives you basically this chunk of the antibody molecule. This is just the V region, and that contains the antigen binding site. And you make the phages make this for you. Look how many different primers you're going to have to use for this. There's like 40 primers here. The reason you need so many primers is because these framework regions, which are not as variable, they still have a lot of variability. Okay, so you're going to need a primer for each of the different mutations that you see here. So they still have mutations, they're just not nearly as many mutations as the CDR regions or the HB regions. Okay, so you're going to need a bunch of different primers, and they are primers that will bind to the framework regions that flank the variable regions of the antibody. Okay, and that will allow you to do PCR to amplify your DNA that encodes the antibody's variable region. Okay, so this is Sanders' slide on this. Step 1, isolate the mRNA from your cells of the mouse that you immunized. Step 2, use reverse transcriptase to turn the mRNA into cDNA, right, complementary DNA. And now you have variable regions for the heavy and light chain. And you're going to amplify only the variable regions for the heavy and light chain by PCR. So these red lines here are portions of the primers that are complementary, and they give rise to the spacer. The spacer is basically a little chunk of DNA that you're going to insert between the variable region of the heavy and light chain. So what you're trying to make is this little tiny antibody that only has the variable regions of the heavy and light chain, and those two regions are linked together by a spacer, sort of like in a little tiny Y-shaped protein. So they actually look kind of like this on the phage. So you're going to stick this DNA that you've made that contains the variable region for the light and heavy chain with the spacer between. You're going to stick this whole chunk of DNA into a bacteriophage, and bacteriophages have DNA as their genome, so you can just splice this right into their genome and then throw it into a phage and infect your E. coli cells with that phage. When new phages are made, they now have the DNA, they now have the protein encoded by that new piece of DNA on their coat at the top of the phage. And now you have the FV of the antibody, you have the entire variable region expressed on the top of the M13 phage. And so a single SCFV is right here, and then there's another one right here. This one only has one SCFV. So after your PCR, you cut and paste into the surface gene of the M13 bacteriophage. You end up with a recombinant phage after you infect the E. coli cells. You have a recombinant phage that replicates in the E. coli. It lyses the E. coli cell, releasing a bunch of phages. And once you recover your phages, you will be able to see that they have the recombinant receptor, single chain fragment variable on the surface of the phage. And you can actually cut off the SCFV little piece right here, and then you can conjugate it to a drug. You can do whatever you want with it. For example, like I said, if you want to target a cancer-associated drug or a cancer-associated antigen, you can make these directed against that cancer antigen and conjugate this little tiny piece of the antibody binding domain to a drug, a cancer drug, and then the antibody, the SCFV molecule will bind to the cancer cell and deliver the cancer drug to that cancer cell. And basically, you have SCFV delivered drug therapy. Okay, and I drew a big long picture of this process with more drawings, right? So you take your antigen in an adjuvant and you immunize your mouse with it. You harvest the spleen and bone marrow, and inside those tissues are plasma cells and B cells that are making mRNA for the light and heavy chains, as well as a bunch of cellular proteins. You're going to first convert all of those mRNA molecules into DNA using reverse transcriptase from HIV. That is a lab reagent. And then this is going to make, the DNA, double-stranded DNA molecule that encodes the light chain and heavy chain of the variable region. So after PCR amplification, in the PCR, you actually have a primer that flanks your variable region of your light and heavy chain, and you have a primer that flanks it the other way. And then you have you've now amplified the light chain variable region and the heavy chain variable region, so you have a bunch of different molecules that are specific for a bunch of different things, but you only have the heavy and light chain at this point, and they're separated. So what you need to do is actually throw a heavy and a light chain together using restriction enzymes. And the restriction enzyme cutting site is located inside the primer. So you're going to use a restriction enzyme to ligate two of these together, right? You might have done this in another one of your classes. You basically create a sticky end and another sticky end, and then they ligate together, and then you use DNA ligase to link these two molecules together. But you're also going to link them with linker DNA in between the two DNA molecules, okay? So any heavy chain present is going to be linked by a linker DNA fragment with any light chain, okay? So you make literally so many different potential combinations of antibody fragments, like billions and billions, maybe even trillions of different unique antibody fragments. And now you actually have, the DNA encoding the heavy chain variable region, and that is linked by linker DNA to the variable region of the light chain, right? And then you splice that into the M13 phage genome, which is a circular DNA molecule, and you do that using restriction enzymes. So now you start with your phage DNA genome, you open that up, and splice in your new SCFV encoding DNA, right? So then you close the DNA, you throw it into a phage, and you let the phages infect E. coli, and they're going to replicate in E. coli, make bunches of phages, and then now the new phages that emerge from the E. coli cell have your single chain fragment variable protein molecule on their surface. And you can pan for the phages that basically are specific for your antigen of interest, and then you wash everything else away. Now you have only phages that are specific for the antigen of interest located on the plate. You elute them from the plate using a glycine buffer, and then you can multiply your phages by infecting more E. coli cells with them. And then once you have enough phages to do something with, you can cleave the single chain fragment variables off and use them for some specific purpose. You can conjugate them to a toxin. You can conjugate them to an anti-cancer drug or some other kind of drug. These can also be used to like treat bacterial infections if you want to conjugate an antibiotic or some other kind of medication to the SCFV. Or you can even conjugate them to a magnetic bead that gets hot in a magnetic field, and it might get hot enough to actually explode a target cell. So a number of different approaches you can use for all these SCFV pieces that you're creating synthetically. So you use this enzyme called papain to cleave off the SCFB molecules from the phage. And then this is just another figure showing you in more cartoonish terms what this looks like. So this is the thing that you're trying to make. The phage looks like this normally, and it's got the coat protein down here, and you want to basically put an antibody fragment on that coat protein. So where does this start? So this starts with taking a gene library from their spleen B cells, turning it into a phage library with PCR, and then splicing it into the phage genome. And then you can pan for the phage library using antigen bound to a plate. And then you wash everything else out, and you're left with isolated high affinity binders, i.e. you're left with antibodies that are specific for your antigen. And then you can infect more bacteria with these phages, grow them up even more, and you can end up with phages that are only specific for the antigen. And then now you have a bunch of different phages, or actually a bunch of the same phage that has the same SCFV on all of their coats, and that is useful because you can conjugate those SCFV molecules to a drug or something else and then cleave it off of the phage and use it and inject it into a patient for treatment. I actually don't know if there's any on the market medications that have used this approach. I would assume there are some, but this is like a pretty involved process, and it's a lot more complicated than the monoclonal antibody. And there's kind of more restricted uses for this because the only reason you'd ever do this is if you wanted an antibody fragment that binds to a little tiny antigen. And for the most part, you don't really need that. But I would assume this approach has been used for certain things. It's probably mostly research stuff. Just immunologists being like, look, I did this. So that is the end of the applied immunology content. So are there any questions for me?
Nope, I'm good.
Okay. Stop the recording.